everyone, welcome back to Microbiome Solutions. Today we are going to discuss about KOH wet mount, which is used for the identification of fungal elements from the clinical sample. Moving on to the principle of KOH wet mount, it is used for the clinical specimen to clear the cellular material for better visualization of fungal elements from the clinical specimen. So KOH preparation is commonly used method for the diagnosis of superficial fungal infection and for the rapid detection of fungal elements in a clinical specimen. As KOH is a strong alkali which separates the fungal elements from intact cells as it digests the protein debris and dissolves the substances that hold the keratinized cells together surrounding the fungi. So the fungi will be trapped inside the keratinized cells as KOH is a strong alkali which will dissolve the cell substances or the protein debris in the cell, which make the fungi more visible under the microscope. So thus the hyphae and conidia or the spores of the fungi can be seen under microscope. So the specimen is placed in a few drops of 10% or 20% of KOH and which is incubated for 5 to 10 minutes and more than 24 hours where tissue sample is used, where gentle heating can clear the samples more quickly. And a cover slip is placed over the KOH digested sample and the slide is examined microscopically without any staining. So different fungal elements like hyphae, pseudohyphae, yeast cells, spores, spirules and sclerotic bodies which can be seen clearly by using a KOH wet mount. And the KOH preparation of sputum, the fungi appear as non-pigmented septate hyphae which is measuring up to 3 to 5 micrometer diameter with a characteristic dichotomous branching and an irregular outline. So we'll discuss about the procedure to make 100 ml of KOH, that is 20% weight by volume solution. So to make that, you need to have KOH pellets, distilled water, a weighing machine to weigh the chemical and a screw cap bottle to store the chemical. So first thing, you have to weigh 20 gram of potassium hydroxide pellets in a weighing balance and transfer the chemical to a screw cap bottle. Then take 50 ml of distilled water and mix until the chemical is completely dissolved. Add the remaining 50 ml of distilled water and make the volume up to 100 ml. And label the bottle and mark it as corrosive. You can store it at room temperature and the reagents will be stable for 2 years. There are two types of KOH method which is done usually in the laboratories. One is slide method and the other one is tube method. We shall see the procedure for slide method now. So for the slide method, the specimens like epidermal scales, nail, hair, skin scraping or tissue has to be kept on a clean glass slide. Pour a drop of 10% KOH on the specimen and you can place a cover slip upon it. And heat the slide gently over the flame so that the protein debris will be detached properly. Leave the slide for 5 to 10 minutes or place the slide in a petri dish or other containers with the lid together with a damp piece of filter paper or cotton wool to prevent the preparation from drying out. So you have to wet the filter paper or cotton wool, whichever you are keeping inside the petri dish, so that it will prevent your KOH preparation from drying out. Examine the slide under the microscope after 10 minutes using 10x and then 40x objectives. So next is the tube test. So for the tube test, we usually use tissue materials or the samples which is difficult to dissolve or difficult to digest using 10% KOH or in lesser time. So for the tube test, you can keep the homogenized tissue material in a test tube and add 20% of potassium hydroxide and incubate the tube overnight at 37 degrees Celsius in an incubator. Following the incubation, you can mix the suspension properly and place a drop of suspension in a clean glass slide and cover it with a cover slip. Examine the slide under the microscope in 10x and 40x objectives. This procedure, which can also be used for nail clippings and skin biopsies, as I have told, which dissolve with difficulty and the concentration of KOH may be increased. So KOH preparation, which is used for the diagnosis of mainly ringworm infection, all the dermatophytic infection, KOH preparation can be used. Laboratory diagnosis of tinea on the identification of an organism by microscopic examination of skin or nail scrapings with 10 to 20% of KOH on wet mount examination. KOH with blue-black ink preparation, which is recommended for Malassezia furfur if it is suspected. And calcofluor white potassium hydroxide preparation can also be used for the examination of fungal infection because calcofluor white is a non-specific stain. An appreciation for fungal element morphology on direct examination is crucial for adequate specimen interpretation because mainly we will be getting clinical specimens for KOH examination and the disease treatment will depend upon the interpretation of the microbiologist. So the limitations of using a potassium hydroxide, as it is a corrosive and deliquescent material, it has to be handled with great care and experience is required since background artifacts are often confusing. Clearing of some specimens like biopsy materials, nail clippings may require extended time. These are the limitations for being using a KOH wet mount in the laboratories in spite of their many advantages. So this is about today's lecture. If you have any doubts, please post it in the comment section. Please do like, share and subscribe to our channel. Thank you.